Good morning. Good morning. It is a pleasure to introduce our presenter today, Dr. Phillip Giampietro, who is a geneticist at the University of Chicago. He will be presenting today about the benefits of genetic evaluation. Please join me in welcoming Dr. Giampietro, to get better, a little better, to the 2024 AMCSI conference. I will be in the back if you need me. Take it away.
Okay, thank you.
Thank you.
Thank you, Jan, and it's a pleasure. I think everyone can hear me. Yeah, no, it's a great pleasure to be here for. I think I started doing this sometime in maybe 2018 or 17, but I really enjoy speaking to people about genetics. It's my passion. So, and sharing some insights that I have and learning your histories and things like that too. So. I'm actually at the University of Illinois, Chicago, but I'm also affiliated with Shriners Hospital as well, where I see patients there too. So I'm going to talk about genetic evaluations and how this could benefit you or your child. I know we have adults here too, so we might want to just consider changing this to me or my child. But genetics, for those of you who might not know, I like to think of genetics as really the science of understanding genetic variation and why are individuals different. And it was all mentioned in the first session that today's Independence Day. People are wearing red, white, and blue. I'm wearing blue and red, and I don't know if I have white on me. Yeah, some white. Yeah. So anyway, I think it's also meant to celebrate our diversity and how we really can understand ourselves, understand each other through diversity as well. But we know individuals are different, and a lot of the differences come in genes that individuals possess and the physical characteristics that emerge as a result of genes. However, genes are not the only story. There's also the environment as well. And many of our traits really are a combination of genetic and environmental factors. And sometimes there's a degree at which genetics are the dominant force or environmental factors can be the denominating force, but also oftentimes it's kind of a mixture of in between types of interaction. So you may have been suggested or referred by your primary care doctor or other doctor to genetics. And what are the reasons that people might seek a genetic consultation? Oftentimes it's to really obtain a more precise diagnosis or for genetic testing, better understand natural history of what a person's genetic condition may be, identify potential treatment options, understand risks for future pregnancies, or what is say my sister or brother may be affected with AMC, and what is the chance for me to have a child with AMC, and how could I best be prepared for having a child, and what things should we do to help make that child's life better. And also helping to work through families' own ideas about causes for their child's condition, also alleviating misconceptions, how things may have happened, how people thought they may have happened, and alleviating parental guilt. And when you speak to people, they often have ideas that they've never shared with anyone about what caused their child to have arthrogryposis, and people really need to kind of sit down with a genetic counselor, geneticist, and also work through this and understand what hereditary is and what does hereditary mean and how changes in our gene, how in our DNA can lead to various conditions. So it's not surprising, and I don't think it's surprising to this audience, that AMC can be caused by a variety of different causes. So when we look at intrinsic, meaning factors relating to our genetic makeup, to our chromosomes, chromosome abnormalities, single gene abnormalities, and Extrinsic could mean also various factors in the environment. So there are different causes, and when we look at there are many different subtypes. There are over 480 genes, genetic factors, individual units of heredity that are known to be associated with arthrogryposis. So that's really rather mind-boggling. And this is work done by Dr. Hall and Dr. Kiefer in various publications that they've found. And they've identified different pathways that affect the central nervous system, muscles, connective tissue, bone, and all genes in these conditions can be at play, and when not functioning properly can result in arthrogryposis. And I think people in the audience know that arthrogryposis is defined. I didn't include that because I didn't want to feel like I was preaching to the choir, but it's congenital contractures. It have to be congenital, okay, by birth, at birth, or noted before, in contractures in two or more body parts. So you need to have the two, and it has to be congenital. Okay, so that's, we don't, for definitions, we do not accept contractures that took place after birth or lately, because those are different processes. Those are more progressive, and that's a different type of what we call pathophysiology, different type of developmental mechanism. Not to say that's not important, they are, but in terms of this audience and what we're discussing and our conversations, we're really looking at congenital. Okay? So, well, yeah, unfortunately, no. I'm sorry that didn't. I think the key things are, when you look at it, what I really wanted to emphasize are arthrogryposis, intrinsic and extrinsic. This kind of gives an array of causes. For the intrinsic, we're looking at the amyoplasia, which is absence of muscle mass. That's 1/3 or so of arthrogryposis, the amyoplasia. But then we're looking at chromosomal connective tissue, nervous system, bone abnormalities. And these are just various examples of that. And then on this side, we're looking at extrinsic. So extrinsic means non-genetic, environmental, differences in the uterus, differences, other types of maternal factors. So these can be infection, for instance, mediated by virus or other various medications, alcohol, myasthenia gravis, and twins. So that's the key takeaways are extrinsic versus intrinsic. I'm sorry it's a little blurry. That's really what I wanted to really emphasize. But, yeah. Right. So that would be more in the central nervous system, structural brain abnormalities, which is a big category. When we think of what can cause us to contract, there's a wide variety of conditions. If you just look at the brain and then go down to the spinal cord and go down to your neuromuscular junction, your connective tissue. Just think of it. This is what we think of, as diagnosticians, and you can do the same thing. It's not. It's pretty, intuitive actually. So let me just hop over here. Okay. So, and that's really what I meant to put here on the top right with nervous system causes, structural brain abnormalities, and that would be where polymicrogyria falls under. Then we look at abnormalities in the neuromuscular junction, like spinal muscular atrophy. Oh, what is this? Here, there's a message that popped up here. Do you want to allow public and private networks to access this app? I don't know. Oh, okay, right, okay. And then various chromosomal conditions. So, okay. Right, so what do we do how many people in this room have had clinical genetics evaluations for themselves or their child? Okay, so I'd say maybe a third to a half. So a lot of people don't know, and maybe this has been something that has been discussed. But the first thing is, you or your doctor or your healthcare provider or your subspecialist or orthopedic surgeon, neurologist, say, well, have you ever seen a geneticist? Would you like to see a geneticist? Would you like to possibly have genetic testing to help understand your condition better and give you more insights, give the medical community treating you. Again, this is not done for research, this is done for you, to help understand your condition and perhaps why this happened, what to expect, what other potential organs we need to maybe worry about, natural history, do we need to worry about hearing, do we need to worry about kidney abnormalities, etc. So that would be the reason, and the process occurs with the referral, and then at the visit, typically it's being seen by a genetic counselor. Genetic counselor is a genetics professional who's had graduate training at the genetics level and to take board exams, as does a clinical geneticist who is an MD and board certified. And we will we'll review your records ahead of time and then make a summary of your medical history, your child's medical history, and then use what we have. Also interview you the day of your visit. We will construct a family history, which is a pedigree, a roadmap of how we can track genes down through a family. Most of the time, when we see families with arthrogryposis, they or their child may represent a single occurrence in their particular family. So we will trace the family out, starting with what we call the proband, the person who is coming for consultation, ask if they have any siblings, what about their parents, are they healthy, etc. What about their parents, do they have siblings, etc. Do their siblings have children? So we go step by step and we construct a vertical and horizontal roadmap of a particular family. We ask about biological relationships. Could parents be related? We call that consanguinity, meaning sharing a common ancestor. And that becomes important if we want to consider recessive conditions where there is a double dose of an altered gene that comes together in an affected individual. We look at other ancillary information, laboratory tests, imaging studies, MRIs, things like that. So we're kind of like detectives and we're data gatherers. We do what we call phenotyping. So phenotype is the outward appearance of somebody, but we need all this information for doing the testing, the complex testing that I'll talk to you about later. And then a genetic counselor will also discuss various psychosocial issues. Where are you living? Who lives with you? Job, school, adjustment, things like that. So peer relationships, etc., which are quite important. Right. So our histories are detailed. So I like to think of myself as a general practitioner, but I have genetic specialization. So we approach cases the way that a primary care provider should. We get a good prenatal history. We ask about, and these are factors that Dr. Hall has identified too in papers that she's written: maternal illness, fever, injury, medications, drugs, alcohol, maternal bleeding. What about ultrasound? Were ultrasounds done? Did ultrasounds show good mobility? As Dr. Hall says, who I quote extensively, is arthrogryposis is all about motion. It's really, is there motion? And because of a lack of motion, things kind of stay fixed. And that's really how you can understand the basis of arthrogryposis. So what did the ultrasound studies show? Was there any prenatal testing, amniocentesis? Nowadays, we can also do non-invasive prenatal testing, which means we obtain a blood sample from mom, and that can be used to detect various aneuploidies, meaning missing chromosomes or extra chromosomes, whole chromosomes as well, and uh, those types of abnormalities, or microarray, which we'll talk about, which can detect chromosome abnormalities below the limit of the light microscope. We ask about weight gain, poly meaning excess, hydramnios fluid, excess amniotic fluid, oligo meaning few, decreased amniotic fluid. How was the baby born? Was the baby born naturally or by cesarean section? And we get all of the various parameters, length, weight, and head circumference. And then we ask if we're talking about a child, we ask about their hospitalization, surgery, developmental history, milestones, motor and speech, personal, social, as well. And we do a complete review of systems, head to toe, and we will then do a detailed family history, which encompasses obtaining family heights, miscarriages, stillbirths, birth defects. We talked about consanguinity, and typically in amyoplasia, although we're uh, um, with the arthrogryposis registry at Shriners, we are looking at many cases of arthrogryposis and including amyoplasia. But now, according to literature and according to papers that have been written by Dr. Hall, for amyoplasia, most of the time, it's an absence of family history of contractures. Okay? So a physical examination typically done by a medical geneticist, but everybody in the room has eyes, and everyone really can be bedside dysmorphologists. And that's dysmorphology is what I studied as a genetics fellow, and DYS means altered, morph is form. So everyone can be an observer, really. And you don't need to go to medical school to be an observer. And parents are the best observers, really, because they're with their child, 24 hours a day, 7 days a week, and they know their child better than anyone. So, and it's often when parents come to see me, they will tell me where every birthmark their child has, and that. So actually, that saves me time when I'm doing an examination because they know where to find things. So parents can be quite helpful. But as part of our exam, we do a lot of measurements. We measure eye distances. We measure the length of the ears, the philtrum, which is this part between the base of your nose and the upper lip. We look for birthmarks. We'll also look at different factors, different parts, including limbs, palm crease. The creases of your palm represent in utero movement. So say if the movement is decreased or your hands are flexed like this, your creases are not going to be as pronounced. And you could also have what's called a transverse crease just because your hands were flexed and they remained flexed. So that can result in a transverse crease. So we like to look at the intensity and the nature of the creases as well. And obviously, contractures of different joints, wrists, the elbows, shoulders, hips, knees, ankles, etc. Foot deformities, dimples over affected joints, and you can see here we've got wrist, we've got actually lower extremity contractures here, and this child has some a chest abnormality here, an indentation in the chest here, or a pectus deformity as well. Some children with arthrogryposis can have what's called a nevus flammus. That's like a birthmark right in the middle above the bridge of the nose, and that is very typical of different types of arthrogryposis, like amyoplasia. We also look at radiographs. What about bone density? What gracile bones? Those can be seen in arthrogryposis as well. And these bones can be osteoporotic or osteopenia, meaning that the bone quality is not very well, it's not very, it's rather weak. And patients can be prone to fractures because of this. So there can be laboratory testing done, and we did a survey of laboratory testing in the literature, myself and Dr. Dieterich, who's a world expert in arthrogryposis in Grenoble, France. And we found that very, very high levels of CPK greater than 10 times normal can be seen in various muscular dystrophies associated with AMC. These are genes involved, or anterior spinal cord disease, and these are genes involved here, or the musculocontracture form of EDS. And there can be intermediate CK levels, neuropathic AMC, or here, TTN, Titan, and some patients with distal arthrogryposis. So CPK is what we call an inexpensive test and can be helpful in some instances of pointing us in the right direction, really. Many of our patients have had EMG and nerve conduction velocity studies, and the yield is up to 100% in neurogenic forms of arthrogryposis and amyoplasia, but drops to 56 or even 34% in myopathic forms. And we view that as more of a second-line investigation. Muscle biopsy, I think most people now are really shying away from muscle biopsy unless there is a clinical suspicion of an underlying congenital myopathy. We don't feel that muscle biopsies are really making an impact in the diagnosis, especially in the distal arthrogryposis group. Then spinal muscular atrophy and Prader-Willi syndrome are really, although they can have contractures in more rarer instances, most of the time they're not really seen in association with arthrogryposis multiplex congenita. So I'm going to go a bit into genetic testing because I think a lot of times this really may prove a little challenging for some people to understand, but I hope I can maybe make it more understandable. And if we start out with some basic vocabulary, right? So human beings have 23 pairs or 46 chromosomes in every cell of their body except the egg and sperm cell, which have 23 or half the number of chromosomes. Okay, so this is a level of organization. We have a chromosome. We have 22,000 about genes, which are distributed on our 46 chromosomes. So these genes make us who we are, really. So here is a chromosome, and then you can see here, we go down to the level of organization of a gene, a unit of heredity. Okay? And genes are made up of DNA or deoxyribonucleic acid. So this is one level of organization here. So I want you to think about, when I was a kid, we had the Encyclopedia Britannica. So I think some people here would be familiar with that, but some younger people might not, because then, we don't make these anymore. We have everything online. But, when I was a kid, someone would knock on our door and they'd want to sell us the Encyclopedia Britannica. And, at that time, that was like a big thing to have the Encyclopedia Britannica, and it had all the information, in the world. And, you had those, and then you kind of knew everything. So, so it was like really neat back then to have it. So here is a whole set of the Encyclopedia Britannica. So that is analogous to all of our chromosomes. So you can see that each chromosome then could represent one whole volume in the encyclopedia, right? And then if you look then, at what is an encyclopedia made of? Well, it's made of different pages that constitute a whole volume. So then we could say that our genes are really like the sentences here on the pages. Okay, so that's what a gene is in this hierarchy. And then what then is DNA? So here are sentences. Well, DNA are then the letters that make up the words of the sentences that are in the volume of the encyclopedia. So you've got then, your you have your gene and you have your chromosome, your whole set of chromosomes here. And this is a DNA sequence. So DNA sequence now is done all automatically, and we go letter by letter. Genes are made up of DNA, and DNA is made up of four what we call nitrogenous bases from biology: adenine, thymine, guanosine, and cytosine. And we've been able to sequence this for decades now, but we've now had over the past over 10 years, maybe 15 or so years, we've had major advances in what we call high throughput sequence analysis, where we can massively parallel sequence hundreds, thousands of genes all at once. So, and that's kind of the testing that we do. So just level of organization, our genome, okay, is everything here in all of our chromosomes, all of our chromosomes. And then we have then here we have genes. Genes are made up of DNA. So get another level of organization. This is resolution. So so this is what we call the haploid genome, meaning 23, not 46. 23 is half the number of 46, and that's called the haploid genome. So you have the sperm and the egg each have half the number of chromosomes to unite and give a total of 46 chromosomes. So the haploid genome is composed of 23 chromosomes. Okay, and then we have, this is the number of base pairs. So this is like 10 to the 9th. A whole chromosome is about 10 to the 8th. And then we could look at bands, which are just little markings on the chromosomes as well, that people in the lab know how to characterize better. We could look at this is all at the microscopic level. So if we took a chromosome and looked at it under the microscope, this is what we would see. Then we get into what you can't see under the microscope, and that's sub-microscopic region here, 10 to the 5th versus, say 10 to the 7th, 10 to the 6th of what you can see under the microscope. So these are sub-microscopic regions. And then we even can go down today to the single nucleotide level when we do sequence analysis. Right. So we can go down to like one base pair. That's what we can do. And that's with what we call whole genome sequence analysis. So we started off with what we call standard karyotyping. Sorry. And then we went into high resolution or stretch chromosomes. Then we looked at microarrays, and I'll show you these in subsequent slides, and whole genome analysis. So we're going to talk about chromosome analysis and microarray analysis. Okay. In the older times, we could only offer chromosome analysis, which was just an ordered array of our chromosomes under the microscope. But in the 2000s, 2010s, we were doing chromosome microarrays where we could then look below the limits of the light microscope and look for more subtle findings, copy number variation, an extra or a missing piece of a chromosome that is not visible under the microscope. Right? And these are the reasons why one would do this. As a general geneticist, I see all types of patients. My practice includes arthrogryposis, but I see children with intellectual disability. A large component is autism, and other conditions like polycystic kidney disease, Turner syndrome, all sorts of other indications, short stature, unusual facial features, abnormal findings on ultrasound, etc. So these are some reasons for performing a microarray analysis. And this is what, now has anyone heard about microarray in this room? Right. So microarray is somewhat of a standard test, but I'm going to say I think genetics practice is going to be even different a few years from now. And we might not even be doing microarray because we may just have whole genome and whole exome, which will kind of cover up and include stuff that microarray is doing. But microarray is a technique that we can use then to look for dosage loss or dosage gain. So we have the patient's DNA that is extracted, and now we can do this just on the cheek swab. So we don't need a blood sample. You just kind of take a swab and put it in rub it in someone's cheek, and then put it in special media and send it off to the lab that you're working with, and that's it. You don't have to send children to get blood tests or anything like that. And it's done, and it's rather painless. So we do this, and we put it in a chip. The lab will stick this in a chip, and you can see this chip has many, many wells. Okay? Has like thousands of them. And they're so small, they're invisible. And each well contains what's called a probe to a particular chromosome region. And what we're looking for is, with that particular region in that patient's DNA sample, is there equal coming together or hybridization? Is there less or dosage loss? Or is there more for that particular chromosome region or dosage gain? And we go region by region by region, and this all is done by a machine, a very sophisticated machine. And here is our chromosome. So this is chromosome number 7. Chromosomes have a short arm or a P arm and a long arm or a Q arm. And we look to see, is there in this region here, this is neutrality right here. Here with a dip is loss of an entire region. That's why it goes down. And here is gain of a particular region. And this is the region right here in question. So you could see, let's just focus on the bottom. There's consistently, say over, maybe 50 or so wells, a dip, okay? And that shows for that region, there is a loss, and that person on that region, one of their chromosomes, it's not present, okay? So that's a depression. So that is a deletion or missing chromosome material. Okay. We can say a duplication is if you have dosage gain. So above is gain, below is loss. So that's my rather, I'm not, the PhD scientist who's reading this, but I'm on the other end, I'm on the receiving end, and these are the reports that come to me and how I have to learn to interpret them. So, but as you'll learn, not all variation is harmful. Some is, some isn't, some we don't know the significance of, and we call that uncertain. So, and this is just another schematic diagram, dosage gain in green and dosage loss in red. And this is simply what a microarray will do, okay, is to look for regions where there's dosage loss or dosage gain. Here is a complement of all of our chromosomes. So this is, again, what the human genome is, 46 chromosomes, 23 pairs, and this would be an array for each chromosome. Notice how all these are pretty neutral here. This is a normal, or there's no extra or missing material, and they're at a neutral position here. Okay, so this person is balanced here. Right? So the diagnostic yield is maybe about 15% or so with a microarray based on the different indications. So let's just talk about what genes are. Genes have a coding region. Remember, genes code for protein, right? So genes are our code. We have the genetic material, and proteins are made. They're not made necessarily all the time. Genes are turned on and genes are turned off at certain times in development. We have a promoter region that is a regulatory region. It kind of says, well, crank this gene up, get ready for it to be made into what we call a messenger. And that messenger then is involved with the making of proteins. And we have our exons and our introns. So the exons are what codes for things. Okay, so the introns are spaces between coding regions, and typically those are taken out. The exons are then kind of joined together. Then DNA is made into RNA, which is then made into protein. Okay, so and we take out whatever is non-coding and then get rid of it. Okay? And this is the DNA code, as we mentioned, a string of the nucleotides here, C, C, and A, code for a specific amino acid, proline, and then this string of C, G, C, code for another amino acid called arginine, and you have DNA that's made into RNA, that's made into protein. You can have then genetic alterations which alter the protein. So that's the testing. And then when we sequence, okay, we are then looking letter by letter by letter to see is the DNA the way it should be with a reference, a human genome reference sequence. We compare what the patient is versus the reference sequence to see if there are differences. Okay, so now this is all done by machines, okay, and it's way past the days of Sanger sequencing, which was very, very long time ago. But that was a step. We couldn't be where we are now without going through the laborious process of Sanger sequencing. And you can get the DNA, you can know what it is, but then how is it read? Like, does this make any sense to you, the way this is now? Like, doesn't make much sense to me. But then you can see that there are words here, and the idea is, what is the frame? How are these read properly to make a protein? What do you start off with? And the machines can kind of pick that up, and here we have what's important then. So you can see the quick brown fox jumped over the lazy dog. The dog lay quietly dreaming of dinner while the dog. So with that, so this is what's meant as the frame, the reading frame. How do these nucleotides, letters, connect to one another to then be read to make a protein? So this is what you need to kind of know, really. So how many people, I'm sure most of you have heard about exome analysis. So this is really, our kind of go-to test in the clinic then. We'll talk about genomes. Genomes not quite there in clinical practice, but more in a research setting. But think of our whole genome as this apple. So genome is the sum of what's coded and what's not coded. So most of what we have in our genome is really information that we just don't understand, really. But the exome is 1% of our genome, so it's this tiny, tiny slice of an apple, and that is about 1%. Okay? And that's what we look at in a whole exome. Okay, so we then we tell families that we may pick up something that is a variation or a change in the genome. There can be normal variation, which don't cause any diseases. There can be disease-causing variation or pathogenic disease-causing changes. There can be disease risk variation, which affect risk of developing genes. And there can be insignificant variations that we just don't know what they mean at the present time. And we then tell families to come back in 2 years for a reanalysis. So back when this was all, coming out, I naively, said, oh, then you just sit down with a book and, you go through each variant and, and figure it out. Well, you can't. You'll be long gone, and you won't even get through a fraction because we have hundreds of thousands of variants, in our genome, and good luck to try to, even figure out, what's significant or non-significant in your own genome. It's really not easy. So we know, as I mentioned before, there's over 480 genes for AMC and about 16 different pathways, which then bring up a whole host of treatment possibilities. This is one complicated pedigree, you can see here, and the squares are males, circles are females, and we have layers of what we call consanguinity, or people having a common ancestor, which are the double bars here. So this is quite complicated, and I give my hats off to whoever was able to ascertain this, because it's not easy to do this. But we have females and males being affected, which is important to note, and we have to work with colleagues to kind of figure out. We're beyond Ps, so Ps now have evolved to genes and genomes and exomes, really. But this is one paper that came up a number of years ago, 7 years ago, where ERGIC1 caused AMC, the neuropathic type. And this is basically what the family phenotype looked like: contractures of the elbows, knees, muscular hypertrophy or enlargement. And then the patient family collected DNA, we meaning the group doing the work, and it was subject to analysis then. And now we use next generation sequence platforms, and these can include panels, although I think now, knowing what we know about the genome, and that we really recommend exomes as a first line test for this. So here is an example of what an exome is. Okay? And we take our genomic DNA, and then we then capture the DNA just for the coding stuff, the DNA that codes for proteins. We get rid of the non-coding. But if we were doing a whole genome, we would not wash this away. We would keep that all. And then we have to sequence, and then we map, we align it. It's rather complicated, but laboratories can do this. Commercial labs can do this, very rapidly. And this just shows here that we will get preliminary results. We then need to review this with our own team. I will get back reports that are about 8 pages or so, and sometimes they say no pathogenic variant identified. Sometimes they will say a variant of unknown significance, and sometimes they will say a pathogenic variant. Now, today, we ask parents, do they want to know secondary findings? So since we're sequencing the whole genome, and that we could come up with inherited cancer susceptibility genes, other cardiomyopathy genes that aren't part of arthrogryposis. And we ask families, do they want to know that information? If they do, we include it. If they don't, then the lab doesn't include it. And do they want to participate in research studies by the lab, et cetera? So those are things that we ask in our sessions as well. And then when we get the report, we then will share this with the genetics team, and we will discuss whether what the lab has identified. If they come up with, oh, we didn't identify any known causative genes, but we have a bunch of what we call variants of unknown significance, then it's my job to look at the variant, to then say, okay, this is what we know about the variant. Does that match what the patient has? So then I have to go look at what the lab says and go to find the papers, and then go say, does this match what the patient has? Sometimes there are things that overlap, and sometimes I can just kind of scratch my head and I can say, well, I don't know, but what does that condition have? What do we need to worry about? And what should the child see a neurologist, a cardiologist, get an ultrasound of the kidneys, etc.? So those are things that we need to weigh in on. And so in this case here, ERG1C1 encodes a cyclic membrane protein, and its function is transport between the ER and the Golgi. Nowadays, we are probably saying that we can get a diagnostic yield of about 30 or so percent, 30, maybe 35% on whole exome sequence analysis. But that's then another 65, 70% that we can't identify a reason. And at this point, I can just say that genetics is changing. We didn't have this over 30 some odd years ago when I was just training as a clinical geneticist. So I'm sure 30 years from now, practice of genetics will be entirely different than what I'm talking about now. So here is a distal arthrogryposis type 1 baby. Baby has this condition here, and there are next generation sequence panels. However, these panels can vary based on the laboratory doing them, and they don't necessarily have all the genes, and we tend to not be doing them these days, really. So you may have heard about whole genome. It's a more expansive test than whole exome. I don't know if in Canada it's available clinically. In the United States, as far as I know, it's not really, or maybe one lab was doing it that can, or there are, but it's more limited in the United States and harder to get approved by insurances. In that case, we're looking at all of the variants. Okay, we're looking at the coding and the non-coding region, and if you could see here, you'll get 3.5 times 10 to the 6 million variants versus 20,000 variants on exome. So no human being can do this, really. That's why we need what's called bioinformatics. So what we do is we take the sample, then we fill out, the genetic counselor will go through my note, and they will get all the salient information, any laboratory tests, what I have in my diagnosis, and they will abstract that, and they will give that to the lab. They will put various codes down. So the lab will then analyze the DNA, and they will filter out genes that the patient doesn't seem to have features related to, but then the lab will kind of look at genes that the patient may have a relationship to and see what variation there is, and then come up with a possible list of 1, 0, 2 or so genes that they think are definite matches, and then they will come up with variants, and then they give us that report and we look at it. So it's not humanly possible to do this without the bioinformatics, and that's a whole new area in genetics, really. People doing that need special training. So if we're looking at research settings for exomes, we're saying about 60% in a research setting in the coding region of genes. But clinically, I think it's probably less than that. So testing today, we can do targeted genes, and that's a specific phenotype. So we're looking at something that's very well defined, and that's kind of in our top course here. So heterogeneity is low, phenotype is very well defined, but now we take a broad approach. We interrogate the whole genome, okay? And so that's very highly heterogeneous and less specific phenotype, but we're able to really interrogate everything. And I think this is the best test that we can really offer. Unless we're convinced someone has a particular condition, then we can go right to that condition and just look at the gene. But most of the time, there's so much overlap at the phenotypic level that you don't necessarily want to bank your money on one particular thing because there's lots of overlap. So this is just showing you, this is one study, one case series done by a group that was actually in France, okay, and they looked in their population, 315 patients, and this is just the frequency of different genes that they saw under exome analysis, and this is the difference of phenotypic features of different organ systems, intellectual disability, bone phenotype, kidney phenotype, eye, hearing abnormalities as well. So you can see that arthrogryposis is a very genetically heterogeneous condition. Like, it's not important to know these genes really, but just the idea that it's very, very genetically heterogeneous and also clinically heterogeneous as well. In this paper here, most of the inheritance was recessive, meaning parents are carriers. They have one working gene, one altered gene, and the child, the affected child, has a double dose, one altered gene from mom and one from dad. They get together and you have a double dose of an altered gene, whereas the next higher frequency was dominant, meaning one single change that is responsible. X chromosome conditions, genes on the X chromosome, when they're altered, will affect themselves more often in males than females because the male has only one X chromosome as well. And the most commonly affected tissue was skeletal muscle here, okay, followed by neuromuscular junction, followed by axonal glial, spinal cord, and brain normality. So there's also research going on into oligo, meaning few genes. So it's not necessarily just one gene, but it can be two genes maybe that interact together to result in arthrogryposis as well. So that's what we mean by oligogenic. And here we can see here this fraction of the pie, about 12% here was associated with oligogenic types of mechanisms for arthrogryposis. And here we had about 40% that was known. And then we can have copy number variants as well, meaning a region of the genome where there's duplication or deletion. So we can solve the puzzle with all of these components as we talked about. History, family history, physical x-rays, laboratory, literature searching. And then we have genetic counseling. So the family will discuss the results with a geneticist and a genetic counselor and discuss the family's understanding of what the results show or what they don't show. And this, like I mentioned, can help the family to cope with better with outcomes and reduce the family's anxiety. And we synthesize all this together and follow up with the family. And some forms of arthrogryposis are not necessarily genetic, such as amyoplasia. And negative information is important for the family. There can be insurance issues, and our genetic counselors are really adept at helping families know what insurance will cover or what they may need to cover as well. So just want to say, genetics has evolved from the time that I was in, high school, and we learned about Mendel's peas and how inheritance worked, and it's not as easy as inheriting stripes and from parents, vertical and horizontal. So there's a whole layer called epigenetics, meaning above the genome, DNA methylation, and that is also important as well. And genetics is an ongoing saga. So what we don't know about today will become known tomorrow. So I always say stay tuned because genetics testing is always evolving. And I just hope I can leave you with this inspiration from Helen Keller. Optimism is the faith that leads to achievement. Nothing can be done without hope and confidence. So, and thank you for inspiring hope and confidence in us as we continue to strive to help discovery and work to serve you better. So, thank you.